Where are we?
I think that was like 40 out of 44.
Okay, so this is going to be like last time. In order for this to make sense and be useful, you need to have your materials. So we'll take a moment, get your lab coats, and get a white rack. The white racks are all back there at the back. And gather up your tubes. They should fit in a white rack. You should have 9 carbohydrate deep, 1 CSI, 1 SIM. Except some of you have more than that, and you have big empty racks. And I moved yours all around. They're not in the racks anymore. Because I had to put them in these pillowcases, so they got in the basket.
Is it an accurate assumption that there's a maximum of 5 right where possible?
Yes. I have to put it on the lid.
Why? No one told Mark Zuckerberg. I didn't know it was on 20
Thanks, man.
They were both on top of one on top of the other.
Oh, why didn't you?
Because I didn't separate the 2 organisms because my fastidious structure is working
It had what?
my fastidious structure thing died right away. So the structure came out as separate place.
No, not yours. Yeah, yours and it's because they had to go in the so you guys put them in and I took them out and put them in a separate rack.
No, this is like final. And travel, and mice, and school, and typing. My thing is though, is like I can read it off of paper out loud. Like I once had a professor here that was like for the final, he said, read them, bring as many pages as you want. Handwrite them or type them. You can write 100 pages of notes if you want. It was a human development class. And it was like you needed 100 pages. See what I did? I read the chapters that the final was on out loud. You bet I did.
That's actually really smart.
She read the what?
She read her notes out loud, so it typed them for her. Was she asleep?
Oh, too bad. I was wondering if you got that. Yeah, I like these ones.
It's interesting that, you know, you have different take on it once you kind of had some experience with this.
Yeah.
And, you know, a lot of people say, well, they're accomplishing this education, and I think they're trying. It's a wonderful balance. Even in this country.
It's interesting that some of these lifted off the bottom.
Gas production.
Yeah.
Yeah. Big time. Yeah, I like it when they split it all apart and they're way up in the air.
I don't even know this kid's guy.
This was like, you must have been studying for anatomy.
So I studied the first half of the study guide, but I didn't realize it's spread onto the second half of the study guide, which most of the test was. So yeah. Sunny, she wrote ouch. She wrote ouch.
Thank you. Yeah. Ooh. Ooh. It's gas. Oh, that was your Oh, we're just saying. So I only
I was telling her, I'm like, I like to read to be ended.
I'll tell you,
Because when I was in Genesis, I read energy. All I saw was energy.
bioenergetics, like, it's my brain, and so she's like, oh, she's I think she understood that. How do you know? I calculate how the grade based on how far away from home. Oh, but I don't know. I don't know. Yeah. I feel like someone's grade is really too high.
I'm gonna refresh the
Not anymore. We'll find you on the progress report. It's like, you know, of course,
Okay.
do you know that other worksheet is like, let's see, right? And I was like, this whole time
Yeah, well.
I am, I'll tell you.
1 point off. 1 point off. For what? I think she should make it. I don't know. Counting my extra.
So if it's off 1.5 off. 1.5.
Oh, right.
Okay, you gotta check my math. I can do it wrong. I'll take it. Okay, so we're going to take a look at the second set of physiological tests. Still working on our physiological unknown. The good news is we should finish these tonight. The other good news is you don't have anything tonight that is fillable. Well, some of you do. Never mind. Some of you do. I'll take that back. I still have some people that have tubes with MRBP medium. Most of you, so those of you that have the MRBP test, the citrate, the urea license control, I'm not going to go back through those again. I'm just going to go through the ones that we are looking at new tonight. So we started out with carbohydrate deets. There were 9 of these. You were supposed to label them as you picked them up because they all had the same pH indicator and they're all supposed to look the same. As I was gathering those up last night, I noticed they don't. So some of them are considerably redder than others. They're supposed to start red and it depends on how they're pH adjusted. Nevertheless, get some of you settled so that people behind you can see. As in, settled. There it is. Okay. So the pH indicator in the carbohydrate deets is always phenol red. You were supposed to inoculate these with your gram-negative fermentative culture, so I hope you did. And you were supposed to inoculate by stabbing them to the bottom. The carbohydrate deets, what we're trying to find out is, can the organisms ferment whatever carbohydrate is in there? And there are 9 different ones. The pH indicator is phenol red. The color change that you can expect to see is shown down here on the bottom, although I've never seen the carbohydrate deets get as pink as the urea auger. So they can produce a little bit of alkaline end product because there is peptone in the medium, but they're not going to produce ammonia. You're not going to see that hot pink color. So the question being asked is, can the culture ferment the carbohydrate? Each one is different. So we have glucose, sucrose, lactose, ribose, mannitol, sorbitol, and inositol. Maybe not in that order. So, just a reminder, these organisms are all heterofermentative. If they ferment the carbohydrate and change the color, we can say they're producing acid. We cannot say it's lactic acid. They're not homofermentative. If they're producing acids, it could be a wide variety of acids. So heterofermentative just means they produce a variety of different end products. They can also produce gas, and some of you may see quite a bit of gas. So when we're looking at the carbohydrate deets, we're looking for 2 types of data. One type of data is color change. If they produce acid, they'll turn the pH indicator, phenol red, from red to yellow. It'd be nice if it was a very clear yellow throughout, and most of them are. Some of our organisms do that. It's like, really? We don't like these tubes that look like rainbows. However, so a couple of things to look at. If you see this kind of result, this medium is pretty much yellow. The only place we see some red is right up on top. And if we look at this culture, we'll find out they have actually spread out across the entire surface. The organisms in air are potentially going to have a respiratory metabolism. They can utilize the peptone and produce a little alkaline end product, but these are definitely fermented. But whatever carbohydrate this is, yes, they're fermenting, they're producing acid. They're also producing gas because this is up off the bottom. This one, I don't think so. I think this is negative. A positive result is yellow. This is orange. So red at the top, orange at the bottom, that's negative for the fermentation. If you've got all 3 colors, like a rainbow, red, orange, yellow,
They're all like that.
So those are really orangey. Again, irritating.
None of mine
I'm
are.
not sure.
They're all the same color.
So that would be
The opposite.
Whoa.
But a lot of mine have gas.
So check this one out for gas production.
Oh, wow.
This is gas production. They move the agar up from the bottom and split it here, so they're really producing.
Yeah.
Well, there's a chunk up here. Oh my goodness. Look at that. It's right there. These guys are producing a tremendous amount of gas. Wow. Gas producers. So, the kind of thing that you would be asked on a lab exam is what two types of data are you looking at here? You're looking at the yellow, which is indication of acid production, and then you're looking at here a break in the auger, or that one where pieces are moved way up the tube. That's indication of gas production. The gas production can be much more subtle. Sometimes you just have a little bubble at the bottom, or you may just have kind of a shiny surface on the stab line where it has been split, and you know the gas just went out that crack and escaped. Does that one have? Yeah, here's one that has just a tiny bit on the bottom, but there's a space down here, so that's gas production. This one, if you roll it, you can also see some bubbles along the stab line. So again, you want to make sure that you record for each different carbohydrate what's going on in there. The ones that are easiest to read are the ones that are distinctly yellow. If they're not distinctly yellow, they're iffy in terms of acid production. Yeah, like this one. This is just irritating. This is bacteria behaving badly. Here.
Okay, I'm calling it a yes.
One thing we might do is look to see what inositol looked like to begin with. So this is inositol. Something looks like it happened in there. It's not red. So hopefully, is this one yours too?
That one is.
That one yours. Okay. Let me find the inositol on here.
She's gonna compare it to the thing.
What do you think about this one?
Yeah. Let's do that. Okay, so this is the inositol medium to start. It's really pink. So by comparison, this is positive. So when these media are made in the back, they do a little bit of pH adjusting. This is toward the alkaline end of neutral, because it's pinker than it ought to be. So this one's positive. So Nikki, you may see the same kind of thing. Is yours inositol too?
Yeah, both of these are.
Yeah, so probably they're positive, because this is the original inositol and it's really pink. Yeah, thank you. Okay, so again, make sure that you do a good job of recording. One thing that you can do is lay them out against a white background and take a picture of them. Make sure the labels are showing so that you know what you've got on each one. Because you want to know acid production and gas production for these different carbohydrates. And again, the kind of thing that you're going to be asked on the lab exam is, what's the evidence for fermentation? The evidence is yellow, the pH indicator changing from red to yellow, and then the split, the breaks in the auger or bubbles in the auger. The yellow color is indicative of acid production. The splits or bubbles are indicative of gas. And we can't get any more specific than that. We can't call it lactic acid, it's just acid. We don't know what kind. The same is true for the gas. Could be CO2, it could be hydrogen. I was noticing that even this tube, notice how we have a slightly pinker top on this. There is peptone in this medium, and if these were in the incubator, the ones that went into the incubator were in 24 hours, but they can still continue to grow at room temp. So I took them out, put them on the box. But if you have a lot of pinkness on top, that's probably from them breaking down the peptone and producing alkaline end products. So gas formation is the crack, acid formation is the color. You want to observe and record for each one. So, we've been talking about the difference between data, result, and conclusion. So for these, the data is color and bubbles. The results are positive or negative. And the conclusion is your culture can or cannot ferment whatever carbohydrate is in that particular tube. This is talking about citrate. We already did this, right? Yeah. So this is kind of a review. Citrate. If they make citrate permeates, they turn it blue. Rest. Not in the right order there, citrate. The urease looks like that. We did that one already. Go ahead. Hang on, this presentation doesn't have everything I want. Okay. Sorry, I had to find a different program. So the next medium that we inoculated was triple sugar iron, or TSI. This has 3 sugars in it. So this is more than one test. We're trying to answer 2 questions, however. One is, can the culture ferment one or more of the sugars present? So this has phenol red as the pH indicator again. If the organisms can ferment and produce acid, they should turn to medium yellow. So the phenol red indicator, red is neutral, yellow is acidic. This medium, however, has iron in it, and so the red is not as clear as the carbohydrate deets. And this test is not as accurate as the carbohydrate deets either. So there's 3 different sugars in here, but we really can't separate them. So the results that you obtain from the fermentation of glucose and sucrose is red in the bottom. That is, you read the results in the bottom of the tube. Unless, of course, they push the thing right out of the tube. Well, if I hold this one up. So this one, the bottom of the tube is yellow. This medium is yellow all the way down. So we would say, yes, these guys can ferment glucose and sucrose according to the results in the bottom of this tube. Does anybody have one that's red on the bottom and yellow up the slant?
No. Mine's like all black on the bottom.
Well, I'll talk about the black in a minute. That's something else. We don't have any that are red on the bottom, or excuse me, yellow on the bottom, red up the slant.
Oh, I do. Yellow on the bottom, red on the slant.
Okay, so this one, the yellow on the bottom suggests that it can ferment, again, glucose and sucrose, but the slant suggests that this culture cannot ferment lactose. You're supposed to read lactose fermentation in the slant, glucose and sucrose in the bottom or the butt.
So even though this one has a tiny bit of red, it's not.
Yeah, ignore that.
Interesting.
I think that's partly because it's dry. So there's 3 sugars, but you can't separate glucose and sucrose fermentation. Glucose and sucrose are together in the bottom, and you can't rely on that slant color either. So this is not nearly as accurate as the individual carbohydrate data.
You said the red indicates lactose.
If it's in the slant, if the slant is red, that indicates they cannot ferment lactose. Each time we're looking at, are they producing acid? The lactose fermentation is supposed to be red in the slant, the glucose and sucrose in the bottom, the butt. But there are some complications with this medium because there's something else going on in here. So this is important to remember. The results obtained from the carbohydrate fermentation in TSI is less accurate than the individual carbohydrate deets. This medium, we used to have these books called DIFCO manuals, and the DIFCO manuals had specific results for TSI for each different organism type, and it had to do with whether that slant was red or not. Sometimes the slant is red, not because the organisms can't ferment lactose, but because they ferment and produce acetylene. If they make acetylene, they're not producing as much acid, and a lot of times the slant will stay red. So it's just know that it's not as accurate as the individual carbohydrate Ds. So in this particular image, the tube that's on the left-hand side, the slant is red. That is supposed to indicate that lactose is not being fermented. So usually a red slant means lactose is not being fermented. If the bottom is black and the slant is red, lactose is not being fermented. That's a negative lactose. If the slant is red and the bottom is yellow, it may or may not mean lactose is fermented. Yeah.
The bottom is yellow and the top, the slant is black.
That is a contaminant. Ignore it. That's all. That always shows up. Sorry about that.
If the black is red and, or the bottom is red and this is yellow, it's not red.
It's not red, it's black.
No, but you said if this is black and this is red, then it's something.
Lactose negative.
If this is yellow.
So that means it can ferment lactose. Again, usually there's an exception to that too. Bottom line is, bottom line for the carbohydrate fermentation, these guys are not very accurate. Well, okay, so there's something else. There's another reason that we use TSI. It's like, okay, if it's not accurate, why do we use it at all? This is just telling you, we do have some that can't ferment lactose, but they can ferment glucose and sucrose, so the slant turns yellow anyway. Yeah. So what we really want to know about with the TSI is, can the culture degrade sulfur-containing amino acids and produce hydrogen sulfide? We're testing for H2S production, and this is significant. The hydrogen sulfide, if they make it, is going to react with iron in the medium, and that's what forms the black precipitate. The black is iron sulfide. If they make hydrogen sulfide, the hydrogen sulfide reacts with the iron, you get iron sulfide, and it's black. So data, black, the result is hydrogen sulfide positive. Except for the one that has the spot way up on the slant, that's just a contaminant, that's not hydrogen sulfide positive. So like this. Yeah, that one's ignore that. That's a contaminant.
Is it coming from the gas pushing the
No.
stuff that
No.
up to the lid?
No, this is a contaminant I can't seem to get rid of, and all it does is turn these dark.
Oh.
If they are producing hydrogen sulfide, the black precipitate is going to be in the bottom because iron sulfide is heavy, so it's black, like agents. Black. That's positive hydrogen sulfide. Okay? And if you have positive hydrogen sulfide and your slant is red, then it's positive H2S, negative lactose fermentation.
This is
If your TSI tube has no black precipitate, then your hydrogen sulfide production is negative. If it's a tiny little speck, it's probably negative. They're not supposed to do that either. What does your SIM tube look like? Yeah, I think it's negative.
Well, it's a little colorful.
Okay, this is a fairly significant test, and sometimes people forget what it is they're supposed to be reading. So again, in TSI, we're most interested in this black precipitate forming. If they form hydrogen sulfide reacts with iron in the medium and produces iron sulfide. It's black. So if you have this black, it's like I said, it's on the bottom. You see this very dark butt on the tube, that means they're producing hydrogen sulfide. This one is hydrogen sulfide positive, lactose negative. This one is hydrogen sulfide, this is lactose positive. This one's lactose negative. They're both hydrogen sulfide positive. Typically a yellow slant above a black butt means lactose is being fermented. A red slant above a black butt means it isn't. As soon as I say that, I think, but we have one culture that will produce hydrogen sulfide and will turn the slant yellow, and it cannot ferment lactose. The good news is you have other ways to determine lactose fermentation. You have a lactose carbohydrate D, and you have McConkey's or T7. You have multiple different ways to determine lactose fermentation. Don't depend on the TSI, it's not accurate. So, we're supposed to be recording data, which is what you see, the colors, and your results, and you're answering these questions. Does the culture ferment glucose or sucrose? Actually, you can't separate them. They're both in the bottom. Does the culture ferment lactose? You read that in the slant. And then does the culture catabolize sulfur-containing amino acids and form hydrogen sulfide? That's the black. If it's black, the answer is yes. If it's not black, the answer is no. Fortunately, there's another way to determine hydrogen sulfide production. So we're not dependent exclusively on TSI. You're also supposed to look at SIM. I'm looking at that and going, wait, where did we skip that one? This is in funny order. Here's sulfur indole motility medium, the SIM. This is the other big tube. So this is going to allow you to ask or answer three things as indicated by the name. S, sulfur. Again, we're looking for hydrogen sulfide production. I is indole, looking for indole production, and M is motility. So again, please don't read this as the SIM test. There is no such thing. This is 3 tests in one medium. So, can the culture degrade sulfur-containing amino acids and produce hydrogen sulfide? If it's black, the answer is yes. If it's not black, the answer is no. So, these are no. Yes. So this is an example of no. There's no black. If you have a positive black on TSI, chances are you also have a positive black on SIM. The other thing that I should mention, if your black precipitate fills the entire bottom of the TSI tube, you really can't read the results for glucose and sucrose fermentation because everything's black. But you have the individual carbohydrate deep, so it's okay. So this is positive H2S, it's black. If there's no black, it's negative H2S. And again, what they're catabolizing to produce hydrogen sulfide is certain amino acids that have sulfur in them. That's just showing you the same thing again. Black precipitate is positive for H2S. So B is positive, A is negative. The next thing we're asking is, can the culture degrade the amino acid tryptophan and produce indole? So indole is a particular end product. Only certain types of bacteria can catabolize tryptophan. If you were going to name an enzyme that would take apart tryptophan, what might you call it? Tryptophanase. That's exactly what it is. So there's an enzyme called tryptophanase that allows them to take apart tryptophan and produce indole. And indole is tested for with a substance called Kovacs reagent. And what you do with it is just put it in the tube. You don't have to worry about aseptic technique because the cultures have already grown. You just put the Kovacs in. So I'll get the reagent bottles out. These are really scruffy looking. The Kovacs reagent is yellow, and what you want to do is put in enough that it is clearly a visible layer on top. Kovacs reagent contains hydrochloric acid, so don't get it on your skin. It also contains amyl alcohol, so it smells funny. And when you put it in, there's this weird precipitate that kind of lifts up. But notice it's yellow. I put it in, it's still yellow. So this is negative for indole production. This culture cannot produce indole. Should we test this one? What's that one? This one is also negative for indole production.
What color was it?
Red. So if you go one more down here. I don't want to test out if it's going to be negative. Sorry.
I'm pretty sure mine's positive. I'm like almost positive mine's positive.
You're almost positive yours is positive?
Yes, I am.
Me too.
You're
Somebody's got to be positive. Sooner or later, we're going to hit one that is. Ah, look at that. She was
Told
positive.
you.
Was positive.
knew it.
How'd you know that?
Intuition.
Okay. So, can you see the color change? Red. And it wasn't long. It doesn't take a long time. When you put this on, you just make a layer that's clearly visible. Don't shake it. You can swirl it around a little bit if you want to, but the SIM medium doesn't have very much agar in it, so it will move a little bit. Also, don't ever lay an SIM tube down on the counter because it will gradually sneak out of there. The medium will come out. So that red color is positive for indole. Having accomplished that, now I can stop. You guys will have to do your own. Okay, so SIM medium, we're testing for hydrogen sulfide, and we're testing for indole production. Again, you put the Kovacs reagent on, you look for the color change. So here's positive indole, red layer. By the way, notice how this has changed color down underneath. If the SIM tubes are allowed to sit for several hours, there will be a change in the color of the agar underneath. Don't count that, because what you want to see is the color change in the Kovacs reagent itself. This, you can see the red going down, but sometimes you'll get kind of a rusty color underneath, even when this is yellow. So yellow in the Kovacs reagent is negative for indole, red in the Kovacs reagent is positive for indole. The third thing that we're testing for in the SIM medium is, can the culture swim? Are they capable of motility? Now, as I said, this medium has a relatively low agar content, so you put them down, you're supposed to stab down and out more or less on a straight line. If they can swim, what do you expect to see? Well, they're going to go everywhere. If they can produce hydrogen sulfide and go everywhere, you get what DJ's got. Black, everywhere. Because they can swim, so they can produce hydrogen sulfide all over. If you look and all you see is cloudiness throughout, they went swimming, and they swim everywhere. So if you can't see the stab line, they are modal, like these guys. This one is an example of pretty good motility. It's just cloudy throughout. You can kind of see where it was stabbed, but they went swimming, and they went swimming all over. So if they swim, they're going to spread throughout the medium. This one I'm not sure. Yeah, if you see the SIM medium itself looks pretty clear, and you can see the stab line very distinctly, that means they're staying right there. They can't swim. I think those are not swimming. I think they're not swimming because the medium is so clear out beyond that. Unfortunately, some of our bacteria are just lazy, and the book says they can swim, but they don't. Like, I'm not sure why they do that. Probably because we keep taking them out of the freezer. So, this is showing an image where the motility is a straight line. I mean, this is lack of motility. See how clear that stab line is? And the medium beside it is clear. So you might need to compare your SIM tube to somebody else's to get a good feeling about whether they're swimming or not. If they swim away from the stab line at all, that's motility positive. If the stab line is very distinct, it's motility negative. Here they just went all over the place.
Mine's just bubbles.
That's actually kind of cool.
Interesting. Well, so this one, you can't see the stab. All you see is bubbles. So I think the reason you can't see the stab is because they actually went swimming. And the bubbles are kind of scattered about. So I think they're modal. Yeah. Bubbles kind of everywhere. Like I said, the black precipitate, it's kind of everywhere. So if the stab line is really clearly visible, that's when they're not modal. If you can't see it because it's black throughout, you can't see it because it's cloudy throughout, or you can't see it because you just can't see anything in there except bubbles.
Yeah, you can't see
They're
the
mobile.
stab lines at all. It's like they cleared it up.
Yeah, I was like, I don't know.
So this says, remember, record the data and results obtained from the SIM medium. This is talking about these little box things that used to be in the laboratory manual. Never mind, I took them out. They were just irritating. People kept trying to put the SIM as one test, and it isn't. So I got rid of those things. Never mind. There's 3 different tests: sulfur, indole, and motility. We already did that. Now somewhere at the bottom of this, here we are. Okay, this says, after you have recorded the results of your carbohydrate beats, your SIM and your TSI, you're supposed to use an identification chart to identify your gram-negative fermentatives. So you should have everything you need to identify, except the chart. So I'll hand those to you.
Page 167.
If it's black, it has hydrogen. It's
No,
hydrogen
don't just start
sulfide
writing on these
positive
randomly, because I want to say a
and
thing or
forms
two
iron
about them.
sulfide. Okay, so basically, if it's not black, then you don't do iron. Yeah.
Yeah, why they did it up there, I don't know.
Just like the part. This is to figure out what you have.
So sometimes they put them
So
on
you
the side.
circle everything.
So if it's yellow, like if these are yellow, then they're
Oh, cool. I've already narrowed it down to 3. So I want the next.
Okay. So, we have across the top the organisms that you can possibly have for your gram-negative fermentative culture. So we have Citrobacter ferundii, Enterobacter aragonese, Enterobacter cloacae, Escherichia coli, Klebsiella pneumoniae, Klebsiella oxytoca, Proteus mirabilis, Proteus vulgaris, Salmonella choleraeus, Serratia marcescens, and Serratia odorifera. There's actually one missing, and I'm going to have to put the data on the board for it. Down the right-hand side, you see our tests, our enzymatic tests, and they are arranged more or less from highest importance to lesser importance. What that means is that your indole, methyl red, Voges-Proskauer, citrate, H2S production, urease, and lysine decarboxylation, those are more important than the individual carbohydrate beat, acid and gas production. But the only one of the carbohydrate beats where we're interested in both acid and gas is glucose. The rest of them, to read this, you're just looking at acid. So indole, again, you're reading that in the SIM medium, don't forget to put your Kovacs reagent on. Methyl red, that was the one where you put the methyl red indicator into your medium after you took the sample out to do the VP test. Voges-Proskauer, that's your VP test, you're testing for acetone. You have to put the Barrett's reagent in. Some of you will have to do that tonight. Citrate, you're looking at color. Blue is positive, green is negative. H2S production, you're reading that both in TSI and SIM. The TSI is supposed to be more significant, but they should be the same. If you've got black in your TSI, you should also have black in your SIM. Urea, well, you're testing for urea hydrolysis. The enzyme is urease. Again, hot pink is positive. Either orange or yellow is negative. The lysine decarboxylation, you have to have 2 tubes. You have to have a control and the lysine tube, and your control has to be yellow or the test is not valid. Motility, you're reading from the SIM. Is your stab line clear or not? And then you have glucose acid, that's the yellow versus red. Glucose gas, that's your bubble. And then you have lactose, sucrose, mannitol, inositol, sorbitol, rhamnose, and rhamnose. Again, you're just looking at acid production. Rather than mark this thing all up, what I recommend you do is record out here where your unknown is, pluses and minuses in this column. Once you have this filled in, then fold this back on the line and just slide this across and look for a match. If you're lucky, it'll match 100%. Sometimes they do, sometimes they don't. They're bacteria, they don't make books. They're notorious for doing what they're not supposed to do. And as I said, there's one missing. I'll put the information
Why
on the
didn't you
board.
just write it in your note results? And then match it from writing all over. It's colored a little.
Yeah, but it's acid negative, and it's not glucose gas doesn't count, so you can record it. It's not gonna be significant, not gonna matter to the identification. That is just the only one.
Put in the drops. When we put the drops in the tube, was this MR? Or was that BP? Was this the methyl red? Or was this rose?
We're supposed to figure that out right now. Pause that. Just pause it, just pause it, just pause it.
This is Rhinella aquatilis. This is Rhinella, and it's missing off the list. That's why it's up here. So this is an option. It's just not on our list. So I apologize. So something's coming up. What do you do next? So after you've recorded the results from all your carbohydrate deeps and your TSI and your SIM, you're going to use the identification chart to try to identify your gram-negative fermentative culture. If it doesn't match 100%, you go with whatever it matches most closely to. If you were working in a diagnostic laboratory, there would be nobody there to tell you if you were right or wrong. Consequently, I won't tell you if you're right or wrong. I can look at your test results and say if I think you're reading something accurately or not, but I don't want to tell you what you have. That's for you to determine. So, yeah, and as I said, the tests toward the top end of the ID chart are more accurate. If you have something that's negative for everything in the top section, that's not right because none of our cultures are negative for everything in that top section. If you think you need to run something over again, you can. Right now, most of the media are back in the refrigerator. So if you have difficulty finding something, let me know. Meanwhile, some students may have conducted some tests or inoculated media that aren't, that we haven't talked about necessarily. So this is just kind of a review of what other kinds of things you might have. So if you had gram-positive cocci, some of them were on mannitol salt agar. If they were on mannitol salt agar or blood agar, either one, you were supposed to run a catalase test. If they were on blood agar, you were supposed to do an esculin test, esculin hydrolysis. If they were on mannitol salt initially, you were supposed to do a coagulase test. So some people had these other tests they were supposed to run. Also, if you had cultures on blood agar, you were supposed to look at hemolysis reactions. So there are a few other tests. This is the catalase test again. You put a little bit of your sample on a slide and you put some 3% hydrogen peroxide on it. Make sure you read the labels on reagent bottles because if you use 3% KOH, potassium hydroxide, it will never bubble like that. The bubble is positive catalase. To get a positive catalase, you must use 3% hydrogen peroxide. A negative catalase is no bubble. The esculin hydrolysis, if your esculin turns black, it's positive for esculin hydrolysis. If it stays tan, kind of a neutral color, it's negative. For the coagulase test, the only people that were supposed to be using coagulase plasma were people that had cultures on mannitol salt agar initially. And you were supposed to take them off of mannitol salt and put them onto TSA, and from there inoculate the coagulase medium. You want to put in a visible amount of bacteria, so you can see that you've put it in there. If the organisms can produce coagulase enzyme, then that liquid will become solid. So solid is positive, liquid is negative. To read it, you just turn them upside down. You don't shake them. If you shake them, you could destroy that solid formation, and then you're going to get a negative every time. So you just turn them over and look at them. Coagulase is solid, coagulase positive is solid, coagulase negative is liquid. This is supposed to be the definitive test for distinguishing between the two species of Staphylococcus that we have. Staphylococcus aureus is coagulase positive, Staphylococcus epidermidis is coagulase negative. On the other hand, you also have mannitol, acid production from mannitol. Staph aureus is positive, Staph epidermidis is negative. The hemolysis reactions on blood agar. I put this information on the board, but we had 3 different types of hemolysis reactions on the 5% sheep blood. If you see a green coloration, and it's kind of a yellowish green, the culture that we have that produces this greening on the blood agar, the colonies are very tiny, and they tend to, when you look at it, it makes the surface of the plate look kind of rough. They're supposed to be alpha hemolytic. They don't do a very good job. It's more of a yellowish color. So alpha hemolysis, the culture is changing the hemoglobin to met hemoglobin, and that's what produces the green color. Again, ours don't do a real good job. If they're beta hemolytic, then the medium clears completely. You hold the plate up to the light, you can see right through it. So the blood cells are gone. They've done complete hemolysis of the blood cells. That's beta hemolysis. And if they do nothing, no change in the color of the blood, that's gamma hemolysis. The gamma hemolytic culture, the colonies will be white, but you'll see no hemolysis on the blood. Now, there were some other cultures that we're messing around with. Some people had gram-negative respiratory cultures. They're either on Mueller-Hinton agar, or they're on TSA. Some people had some fastidious cultures. We had gram-negative on brain-heart agar. We had some gram-positive on brain-heart agar. We also had some gram-positive on nutrient agar. So I'm going to just go through quickly how do you identify specific genera in these major groups. So we're back to group 1 gram-positive cocci. If the catalase test is positive, how do you know? Again, that's hydrogen peroxide. If it bubbles, it's positive. You want to know, does the culture ferment mannitol? Look at your mannitol salt plate. If it's positive for mannitol fermentation, the plate will be yellow. Then you want to see, can it ferment glucose and form both acid and acetoin? To test for acetoin, that's the VP test, so you run an MRVP. If you had staph, you were supposed to run an MRVP. And then the last thing was the coagulase test. You had to inoculate the coagulase plasma. So if you had a culture that was gram-positive cocci, that are catalase positive, they ferment mannitol, they ferment glucose, forming both acid and acetoin. So you're going to have a positive MR and a positive VP, and they are coagulase positive. They're identified as Staphylococcus aureus. So you would follow your dichotomous key and you would get to this point. If your culture is gram-positive, catalase positive, but they can't ferment mannitol, so the mannitol salt plate stays pink, you put them into glucose for the MRVP. The references we have say that they are positive for both, but ours aren't. They really don't do a good job. They're kind of weak at fermentation of glucose. They will usually test positive on the MR, very rarely test positive on the VP, and they are coagulase negative. If they do that, they're Staphylococcus epidermidis. The staph epi will typically leave that mannitol salt plate pink, and they really don't do a good job of fermentation in the MRVP. Now, if you have gram-positive cocci that are catalase negative, these are our fastidious gram-positive cocci. You were supposed to inoculate esculin, and you were supposed to look at the hemolysis reactions. So if they hydrolyze esculin, the medium turns black. And on the blood plate, they're gamma hemolytic, so there's no change in the color of the blood. They're identified as Enterococcus fecalis. These used to be categorized as Streptococcus, and their genus name got changed. They're now Enterococcus. So Enterococcus fecalis. If your culture is catalase negative, doesn't hydrolyze esculin. There's no reaction on the esculin medium, and they are beta hemolytic. In other words, your blood plate turns clear. They're identified as Streptococcus pyogenes. And if you have a culture that is catalase negative, they can't hydrolyze esculin, and they are alpha hemolytic, the medium turns greenish, they are Streptococcus oralis. So we have two different Streptococcus, one Enterococcus, two different species of Staph. Okay, now we're looking at group 2, the gram-positive bacilli. They should be growing on nutrient agar, although we do have one gram-positive bacillus, actually they're kind of pleomorphic, that are fastidious. We know the culture's not obligately anaerobic. How do we know that? None of our cultures are obligately anaerobic. Why not? They're growing in the room air. The room air has oxygen in it. We know because we need it to survive, and we're breathing. So none of these guys are obligately anaerobic. So if you have gram-positive gram-positive bacilli on nutrient agar, you want to know if they're making endospores. Do you have to do an endospore stain? No. You should be able to see endospores just fine with a gram stain. Endospores will show up as white spots in a gram stain. So then you want to know, where are those endospores and what shape are they? So just look at your gram stain. If when you first are working with your culture, if they're too young to have made endospores, just go back and stain them again a couple of days later. Just do another gram stain. Anyway, so you look at the shape and location of the spore, and is the sporangium swollen? The other thing we want to know about these gram-positive bacilli on nutrient agar is how big are they? We want to know what's the diameter, and we also want to know, do they form chains or not? If they form chains, they're streptobacilli, and then, so it's just morphology that we're using to identify these guys that are on nutrient agar. So if your gram-positive bacilli on nutrient agar are not obligately anaerobic, as I said, none of them are, they form endospores that are central and ellipsoidal, so they're much longer than they are wide, and the sporangium is not swollen, but the cells are in chains. They form streptobacilli, and their diameter is greater than 1 micron. One other thing, what is the stain that you should use to measure? The indirect. So everybody should do both a gram stain and an indirect for their physiological unknown 1 cultures. The people that have gram-positive bacilli have to, because they have to measure. But because they have to measure, you all have to measure. It's a requirement. If they are greater than 1 micron in diameter and they're in chains, they are Bacillus cereus. These are pretty big organisms. If they are not obligately anaerobic, they form endospores that are central and ellipsoidal, sporangium's not swollen, but the cells are not in chains, and they're little. They're less than 1 micron in diameter. They're Bacillus subtilis. Bacillus subtilis are significantly smaller. And then if they're not obligately anaerobic and they form spores, but their spores are terminal and typically spherical, so they look like balls instead of jelly beans, and the sporangium is swollen. So your cell will look like a tennis racket. You have a cell with a big ball on the end of it. They form single cell arrangement, and their diameter is less than 1 micron. They're identified as Bacillus psychodurans. These guys don't grow well in the incubator either. They're very slow. Then we have our group 2 gram-positive bacilli that are on brain heart agar. Not very many people have the gram-positive bacilli on brain heart. Again, they're not obligately anaerobic because nothing we work with is. And they do not form endospores. They are pleomorphic, however, so their shape is quite variable. They can be sometimes cocci, sometimes bacilli, and sometimes branching bacilli. They can form cells that are a Y shape. So if you have branching bacilli, beware. But just having gram-positive organisms on brain-heart auger is significant. Our brain-heart auger is for fastidious microbes. There is one test that you're supposed to use here. We were depending primarily on the Y shape. Then we found out these cultures are not very cooperative. They don't always produce the Y shape. So then we added to this the nitrate reduction test. The nitrate reduction test, you have that? No? Didn't I give you one of those? Is that it? Yeah, that's it. Oh, you put Kovac reagent on it. That's not good. Oh, I did?
Yeah.
Oh, gosh, I did.
Yeah,
I really
so-
messed you up. You're gonna have to do this over again. Sorry. I thought it was your, sorry. The nitrate reduction. That's bad. So this is a medium that we're testing to see can the organisms use nitrate as a final electron acceptor. If they can, then they can reduce nitrate, and they reduce it to nitrite, which we test for using two reagents that have to go on top. Like, sorry. Anyway, so you'll have to inoculate this again. The reagents that go on here are nitrate reagent A and nitrate reagent B. So the only people that had to run this test are the people that have gram-positive fastidious organisms. They're on brain heart, and they're kind of strange. And this is not a test that you're going to be tested on. It's just useful for the identification of these guys. And the other reason for introducing is because it's also very useful for the identification of semester project organisms. A lot of our semester project organisms, this test is significant. So we decided to go ahead and set it up. And I messed it up for you. I'll have to get you another one. Sorry. Anyway, if they do that, they are identified as Raphia denticarius. So you can actually identify these just by looking at the ground stain and seeing those Y-shaped cells. Then we're into group 3-4, the gram-negatives. You had to determine if they were respiratory or oxidative, respiratory or fermentative. Respiratory, oxidative, same thing. What medium did we use? OF, oxidation fermentation medium. And then we want to know, do they form cytochrome C? That's rub them into the filter paper on the little plate that's up here at the front. We're using the oxidase test. And then we want to know, is the culture fastidious? How do you know if they're fastidious?
Look on the blood auger.
They're either growing on blood auger or brain heart auger. They're on brain heart, but fastidious. So, if your gram-negatives are mostly cocci in shape, they're respiratory, so that your OF medium is green under the Vaspar seal, and they do produce some aerobic acid, so the top of the unsealed tube turns yellow, but when you rub them into the filter paper, they do not produce a purple spot. So they do not produce cytochrome C. They're oxidase negative. They're identified as Acinetobacter baumannii. If your culture is mostly bacilli in shape rather than cocci, they are respiratory, but they do form cytochrome C, so your oxidase test is positive, and the culture produces a pigment called piocyanin that is water-soluble, goes into the medium. Then they are identified as Pseudomonas aeruginosa. Some of you could identify that one just by the green color that went into the medium. And they smell funny. I think they smell like grape bubble gum. Other people think they smell like tortilla chips.
Very
I don't get the combination. It's like, really? Tortilla chips? If they are mostly bacilli and they're respiratory, but they produce alkaline end products, so the open tube in the OF is blue. They form cytochrome C, so you rub them into the filter paper and they make a purple spot, but they don't form any green water-soluble pigment. Then they're identified as alkaligenase fetacalis. The blue color you see on the lettering there is about the same color as the blue color in the OF tube, the open OF. So then if they're mostly coxy and they ferment, but you don't know they ferment because they're fastidious and they won't grow in anything other than our medium that is enriched medium. So they're growing on the brain heart. The colonies are kind of tan-like. Anyway, gram-positive coxy in pairs. You're identified as Neisseria lactamica. I think that's all the different organisms that we have. So what are you supposed to do? That, if you've gotten to this point, then the investigative portion of your physiological unknown one is done, or very close to that. We still have a couple of people like Sonny who I messed up his test, so I have to run it over again. Oops. So what are you supposed to do? You're going to do a write-up. Notice it says short write-up. This is because I don't really want something that's 20 or 30 pages long, but this is going to be a complete summary of what you did to identify your culture. It's going to include cell morphology as determined from a gram stain and an indirect. So you remember that little section where you were supposed to write in paragraph what your cells were like for the morphological unknown? You get to do that again for the physiological unknown, except you don't do acid fast or endospore stain or capsule stain. It's just indirect stain, which gives you size, shape, and arrangement, and gram stain, which gives you what's their wall like. You can run a KOH test also to double check your gram stain. So cell morphology, including gram stain and indirect, colony morphology from your plate medium. You have two different cultures. One of the media that you use should have been T7 or McConkey's. So remember, for your colony morphology, don't forget color. Color of the colonies, color of the medium, because it tells you, do they ferment lactose or not? And that's part of your physiological test. So you have to put that in there. Otherwise, your colony morphology is going to be form, margin, elevation, surface texture, optical character, all that stuff. Don't forget size. Size in millimeters for colonies, size in microns for cells. If you get a color change on a selective and differential medium, include what that tells you. They're yellow on mannitol salt agar, that means they ferment mannitol. If they're hot pink on McConkey's, that means they ferment lactose. So you want to put that in there. Then you're going to give an explanation of each test that was required for the identification, and this is what you need to know. The name of the test, for example, the Voges-Proskauer test. Second thing is, how does the test work? Well, for the Voges-Proskauer test, it required that you take a milliliter of the culture and put it into a screw top tube, add 18 drops of Barrett's reagent A, 18 drops of Barrett's reagent B, shake it for a minute, then let it rest 10 to 15 minutes, then shake it again. And then your data, what did you see? Well, it turned kind of wine red or pink. Actually looks like raspberry Kool-Aid. So you see pink, the result is positive, and the conclusion is this culture can ferment glucose and produce acetoin. So in other words, you're going to analyze what each one of these tests is telling you. Name the test. Explain how it works. If there's a pH indicator, name it. Can you what? You can make a chart, but the reason I don't give you the chart with the boxes is because the boxes are too tight and you tend to leave things out. So I want to know if there's a pH indicator in there, if it's citrate utilization. The pH indicator is bromothionyl blue. And then tell me how it works. If it's green, it's neutral. If it turns yellow, it's acidic. If it turns blue, it's alkaline. So the pH indicator and how that indicator works. Because basically what you're building here is a study guide for you for Lab Exam 2. You can use pictures. Pictures are great. So where you're using a pH indicator, say here's our pH indicator, phenol red. Red is neutral, yellow is acidic, hot pink is alkaline. Just put the colors on there so that you can, especially if you're a visual learner, you want to have those pictures because it helps you learn. And for Lab Exam 2, you're going to have tubes out there on the bench surfaces. You're going to be looking at the tubes. So you need to know what's going on. The conclusion in each case is, what does that test tell you? The culture can ferment glucose and produce acetylene, or the culture can produce the enzyme citrate permease and pull citrate into the cell through the membrane. But I don't want it to be as long as the descriptions that are in the lab manual. Try to cut it down if you can. Shorten it a bit. You have to identify 2 organisms, so make sure you identify both. Genus and species names required. And so keep it short, as in don't just copy the lab manual word for word, please. If you use boxes, make sure you complete it. Okay? It's worth 50 points. When is it due? It's due when we finish the DNA exercises in lab. That's a ways out. So it's way past lab exam 1, which is good because you need to do that over the weekend. Study for lab exam 1 over the weekend. Okay? Again, the identification using the chart, you go with what matches best. And then see if you can get it to come out the way it's supposed to. If you get it wrong, it's only 1 point.
We write, I know we have to identify 2 organisms. We write 1 before each organism.
Pretty much.
Okay, so we do. So okay.
Now another question that nobody asked, but I'm going to answer it anyway. Please type it. I want it typed. The word processor's fine. I don't want these handwritten. Please type them. Word processor, whatever.
When you say short, how short is short? Like, we make it like as long as we want?
Pretty much.
Okay.
Especially if you put in pictures.
Okay, because mine's not gonna be short.
You're not going to get dot points if they're long.
Okay.
That's fine. Did I announce that?
No!
Oh.
Alright, Gabe, you can stop it now.